Good morning, everybody. My name is Josselin Ortiz, and I'm a biomedical technician with PBL, which is a CRO that's owned by Cedars-Sinai. So this is my presentation on high-throughput and automated cellular and tissue lysis using Covaris AFA technology for proteomics. And this presentation will hopefully give a little bit of insight as to why we decided to incorporate Covaris technology into our cell and tissue lysis workflows. In this presentation, I'll also share some of our astral data that we achieved through our cell and lysis experiments. So if you're interested in reading more, there's a QR code on the screen that you can scan to access it. So this is a table of contents just to kind of outline all the main talking points in my presentation. So first, I'll give a little bit of a brief overview with some key background information. After that, I have our cell and tissue lysis workflows and the results that we were able to achieve there. Following that, I have a section for recommendations that I would like to give based on our experience, and then just a conclusion to summarize everything. And so, as I mentioned, we're going to begin with the overview section where I'll give some key information and hopefully give a little bit of understanding of how we came to the conclusion to include Covaris into our workflows. So I just want to begin by giving some key ideas. So as many of you know, sample preparation is probably the most important step in any workflow, especially when you're limited by your starting material. Reproducibility is a key measure for locking in a workflow. A variety of different sample preparation methods have been used to generate protein expression profiles for many different types of cells and tissues. And lastly, the biggest hurdles that we face are low reproducibility, proneness to error, and throughput issues. So this image here is really just to illustrate the different methods of sample preparations used in different proteomic workflows. Some of these methods our group incorporates into our workflows, but others we don't. But it's really here just to highlight the versatility and the different approaches that you can take. So as you saw on the last slide, it can be kind of overwhelming to know where to start. And so when we were tasked with lysing cells and tissues, we knew we needed to lock in a method, but we just didn't know what that method was going to be initially. And so we tried a couple of different methods, and this is what we observed. So first, we tried a physical lysis. However, we found it to not be high throughput or automated, and we had issues surrounding reproducibility. Next, we tried a protein precipitation method. However, we had longer incubation times. We had more areas for error to occur, not high throughput, automated, and we had issues with pellet solubilization. And lastly, we tried Covaris, and we found it to be more versatile, easier to use. It had the capability of being high throughput and automated. We found good reproducibility, and we also had a good partnership with Covaris, where like Samir was really able to help us figure out methods and just answer any kind of general questions or issues that we had. So we definitely saw the most possibility of success with Covaris. However, because Covaris' AFA technology addresses the bottleneck between different sample types and downstream applications, it utilizes focused acoustic energy to precisely and efficiently fragment samples without the introduction of heat or bias. And so as I mentioned, AFA technology delivers a controlled dose of acoustic energy to the samples without human contact. And it does this a column at a time. And we're a group that does a lot of high throughput work. But being as this was going to be our first time or first go around of using this system to lyse our cells and tissue, we knew we were going to have to do a lot of experimentations. And so we really needed a system that could do a high throughput, like a whole plate of 96 samples, but we also needed to do a lot of smaller scale experiments to really fine tune our workflows. So now that I got the overview section out of the way, we can begin with our workflows for our cell lysis. So this is kind of an overview of the steps in our cell and tissue workflow experiments, and you can see that they're very similar, but for right now, I'll be focusing on the cells. So for our cell lysis, we did our experimentations using HEC293 cells. and we would resuspend them in our lysis buffer, and depending on the cell population amounts, you would either do the strips or the MT130s, but regardless, you would resuspend our cells in our lysis buffer, and then we would place them on our Covaris LE220-plus instrument and do a BCA to determine protein estimation, and then once that was completed, we would be able to do our automated SP3 digest. And once the digest was complete, we would place them on our Astral, and they would be analyzed using Diane in a perfect environment and library-free mode. So knowing that we were moving forward with Covaris to lyse our cells, we had to perform a reproducibility experiment to determine whether we could achieve consistent results. And so we conducted a 3-day experiment using the strips with varying amounts of HEC293 cell populations in order to ensure reproducibility. And we tested 4 different cell populations. So we did 250,000, 500,000, 750,000 cells, and then 1 million cells. And we were interested in seeing whether we could reproduce a linear trend throughout all 3 days. And we did in fact see a reproducible linear trend, but in conjunction with that, we were interested in seeing if we were to extend it beyond 1 million cells, would we still see that reproducible linear trend? And so we did a smaller scale experiment using 4 replicates of 2 million cells with our MT130s, and we did in fact see that the reproducible linear trend continued. And so, as I mentioned, we did our reproducibility experiments, but here are the results from our BCAs. So we know that a typical yield across different cell types varies between 100 to 350 micrograms per 1 million cells. And based on this, we do see that the reproducible linear trend in terms of protein yield is increased with the number of cells. And so this heat map here shows the results of our BCA. And so we saw for like 250 cells as our starting cell population. If you look at the scale here, you can kind of see what our results were. So for this one, it was around 25 micrograms, and you can see that the color is pretty consistent throughout the different days, and that holds true for the other cell populations. So for 500,000, it was around 50. So we basically got around a 10% recovery for our experiments. So here are the LC-MS analysis results from our 1 million cells from our reproducibility study using Astral. So this was run on a 24-minute gradient, and we identified around 8,000 proteins with pretty low miscleavages, which really highlights the efficiency of our automated SP3 digest. So now that we have cells out of the way, we can move on to the second part of our workflows, which is our tissues. So this is the same overview that I showed before, except this time now I'll be focusing on our tissues. And so for tissues, we began by placing our desired amount of tissues into our lysis buffer. And again, we placed it into our Covaris LE220-plus instruments and did a BCA to determine our protein estimations. And once that was estimated, we then digested it using our SP3 automated method. Then they were prepped and ran on our Astro and ultimately analyzed with a Diane in a perfect environment and library-free mode. So similarly to how we did with cells, we also wanted to conduct a reproducibility experiment for tissues. So the Covaris team was really helpful, and they really brainstormed with us and helped us provided us with recommended methods, and even provided us with MT130s, but at the time were a prototype product. So we conducted a 3-day experiment for each tissue type and used the MT130s to lyse the tissues. We usually use between 3 to 5 milligrams of tissue in 120 microliters of lysis buffer. And so far, we've done a reproducibility experiment with 4 different tissue types, and they're intestine, liver, lung, and heart. And so this is just a comparison slide that really highlights the consistency of Covaris. So here's a comparison between different tissue inputs and its associated BCA protein yields for 2 different taxonomies. And so the comparison of protein yields was more or less similar, irrespective of the starting input material or the taxonomy. And here are the intraday protein ID summary results, where you can see the numbers that we achieved from our reproducibility experiment for each tissue type when ran on a 24-minute gradient on Astral. And for heart, we identified around 6,800 proteins. For intestine and liver, we identified around 10,000 proteins. And for liver, it was around 8,000 proteins. And so, as I previously mentioned, one of the biggest hurdles that we can face is reproducibility, but it's also one of the key measures for locking in a developed workflow. And so we really wanted to make sure that our numbers didn't just look good, but that our numbers were reproducible across the different days. And we did in fact find that to be the case. So all of our identified proteome had CV values below 30%, and most were even below 20%. So we were also interested in seeing what the unique proteins of each tissue type was, as well as the overlap across the different tissue types. And so this Venn diagram illustrates just that. So 1,007 proteins were unique to lung, 252 were unique to heart, 403 were unique to liver, and 980 were unique to intestines, and 5,949 proteins were shared across the different tissue types. So around 47% of proteins are common across the different tissue types. And then based on that previous Venn diagram, we took the unique proteins from each tissue type and did a functional analysis using a molecular signature database to see which canonical pathways were enriched. So if you look at this dot plot where the dot size represents the number of proteins associated in the specific pathway, and the color of the dots represents FDR significance. So based on the results of the functional analysis, our results suggest that we could see tissue-specific enrichments of our pathways. And so just to highlight a few, for cardiac conduction, that's a pathway that is associated with the heart. Metabolism of lipids, that's a pathway that's associated with the liver. Digestion and absorption, that's a pathway that's associated with the intestines. And lung fibrosis is something that's associated with the lung, sorry. And so now that we have the workflows out of the way, we are now moving on to the recommendations that I have based on our user experience. So due to Covaris' versatility, we were able to use our own Lysis buffer. And so the first hurdle that we experienced was when we started experimenting with cells. And so we noticed that when we were resuspending our cells in our Lysis buffer, it became viscous immediately. And so to combat that issue, what we did is we started resuspending our cells in a detergent-free solution, and then just adding SDS to it until we have our desired concentration. The next issue that we had was when we started working with tissues and we started using the MT130s. We initially were weighing our sample into Eppendorf tubes and then transferring them to the MT130s. However, we experienced a lot of sample loss, and it was just very difficult to transfer sometimes, especially when the tissue started to defrost. And so the way that we got around that issue is we started placing our MT130s into our Eppendorf tubes, and that really minimized the transfer steps, and therefore it really minimized sample loss. The next recommendation that I have is to use either disposable biopsy punchers or scalpels and find what's the easiest way for you to handle the tissue. For me personally, I like using a scalpel better. But one thing that really helped with that is that we got a dissecting kit, and that really helped with the tissue cutting process. One thing that we didn't know at the beginning, but now it makes sense as to why we were having issues with our Lysis in the beginning, was that we were not degassing our system properly. We thought it was just kind of like, oh, you can skip it if you want, but you really can't. So just make sure you really degas your instrument every single time you use it. And similarly, we found that it's best to just leave the Covaris in service mode. Your instrument's not going to die, or the system's not going to die if you don't do it, but just try to do it if you can. And because of Covaris' versatility, there are many different parameter settings that you can play with. Some samples require more time or less time, so the versatility allows you for really to play with those settings. And similarly, if you're having issues with methods or something, it's always a good idea to reach out to Covaris and ask them for help. They've always been more than willing to help us with any issue that we've had, and so they've been a really helpful resource. So now that we have the recommendations out of the way, we can now move on to the conclusion. So in conclusion, reproducible linear trends were seen with increasing cell numbers based on our BCA. Using Covaris in conjunction with our automated SP3 digest, we were able to identify over 8,000 proteins on Astro for cellular proteomics. Our results closely mirrored the figures that were, our results were similar to the results that Covaris saw in their internal research into their internal research. And expanding our investigation to different tissue types, we employed Covaris in conjunction with our SP3 digest. We saw a range spanning from 6,000 to 10,000 proteins on Astro on only a 24-minute gradient. And tissue-specific proteome pathways enrichment was observed, and lastly, Covaris, we found it to be high throughput, reproducible, and versatile. So thank you, everybody, to my team, to PBL, everybody at Covaris. Everyone's been super great. Thank you.